So in today's talk, I'll be talking to you on artificial enzymes for the regulation of redox signaling. I thank the ACS for organizing this science talk virtual lecture series. Before I go to our work, I would like to briefly talk about enzymes. And I'm sure all of you know about enzymes. They are biological molecules, typically proteins. They speed up chemical reactions that support life. And that's the reason we call them, biological catalysts. They play essential role in various biological processes, including respiration, digestion of food, nerve functions, muscle functions, and many other functions. They bind to molecules and alter them in very specific ways. And I'm going to talk about some of the very, very specific examples of how they alter specific ways. The human body contains hundreds of such enzymes. And some of the very, very basic enzymes that we know, for example, amylase that converts starch into sugars, and as you know, glucose, the most important source of energy in all organisms. And this enzyme is present in saliva, and that begins the chemical process of digestion. So there are various other enzymes. I'm not going to talk about the classifications of enzymes. I will restrict myself to one specific enzyme known as redox enzymes, a class of enzymes known as redox enzymes. So my talk is going to be on artificial enzymes. You might have heard this term, artificial enzymes. So the synthetic molecules that functionally mimic these enzymes. We know enzymes are very specific. They convert substrate to products through enzyme substrate complex, and they catalyze the reaction in the active site of an enzyme. So for example, one of the active site I have shown here, these are metalloproteins heme active site. And the chemist tried to mimic the function of this enzyme using small molecules, synthetic enzymes. And the synthetic biology yielded catalysts from, for example, synthetic genetic polymers. And in 2015, artificial enzymes have been generated using synthetic genetic polymers. And what is challenging is that, can we design an artificial enzyme that can perform specific reactions inside living cells? And chemists, we try to do such enzymatic reactions outside the cells, and we show that they are efficient. Some of them use the active site similar to the enzymes to perform such reactions. So the aim of our study is to design artificial enzymes to regulate metabolic, cardiovascular, and neurological processes under disease conditions. So I'm not going to talk about the proteins that act as artificial enzymes. I will restrict myself in this talk to talk about small molecules that function as enzymes outside the cells and also inside the cells. And one particular class of compounds we are interested are antioxidant redox enzymes. And many of you are aware of these antioxidant redox enzymes. There is an enzyme known as superoxide dismutase, which is present in mammalian cells, which converts superoxide to hydrogen peroxide. Once hydrogen peroxide is generated, two enzymes act on that: catalase, a metalloprotein, which convert hydrogen peroxide to water and molecular oxygen, and the glutathione peroxidase, a selenocysteine containing enzyme that converts hydrogen peroxide to water without generating any molecular oxygen. Before going to our work, I will talk about the role of hydrogen peroxide and the central redox theory for aerobic life. Humboldt synthesized one of the first synthetic peroxides, barium peroxide, in 1799 as a byproduct of his attempts to decompose air. In 1818, Theonard first described that the treatment of barium peroxide with nitric oxide produced a compound which he described as oxygenated water, and now that is termed as hydrogen peroxide. So the first indication that hydrogen peroxide was formed in vivo followed from the discovery of catalase by Louis 119 years ago in tobacco leaves. And this actually set the initial experiments on hydrogen peroxide. A paper titled A New Enzyme of General Occurrence in Organisms, and then role of hydrogen peroxide in cellular respiration was studied. However, the role of this small molecule, hydrogen peroxide, was a matter of intense debate between Heinrich Wieland and Otto Warburg in 1920s. And Wieland proposed the formation of hydrogen peroxide during respiration. But Warburg said, because there is no experiment that supports the theory and no consequence that fits the theory, it should no longer be taken into account. And a lot of intense debate between these two people, and that led to Wieland to sacrifice a dog, but he did not find any hydrogen peroxide. And today we know why it's difficult to find hydrogen peroxide, because number of enzymes act on hydrogen peroxide, use hydrogen peroxide as the substrates. Hydrogen peroxide and the central redox theory for aerobic life involves conversion of molecular oxygen to hydrogen peroxide and then all the way to water. If we add one electron to molecular oxygen, we get superoxide, and which is produced by NADPH oxidases, xanthine oxidase, and so on. If you add one more electron to superoxide, we get peroxide, hydrogen peroxide. One more electron cleaves this species to produce hydroxyl radical and water, and the fourth electron converts this to water. So when oxygen goes to water, 4 electron reduction process is involved, and all the species that we see in between called partially reduced oxygen species are reactive oxygen species. Superoxide, hydrogen peroxide, and hydroxyl radical, the 3 major reactive oxygen species in the human body that controls number of biological processes. If you look at the superoxide, which is controlled by an enzyme called superoxide dismutase, various isoforms of superoxide dismutases are present in mammalian systems. So the major one is SOD1, which is present in the cytosol of a cell, and SOD2 is a manganese containing superoxide dismutase, which is present in mitochondria. But the basic function of the superoxide dismutases is the conversion of superoxide to hydrogen peroxide and molecular oxygen. It can use copper, it can use manganese, iron, and it can use nickel and various species. But what is the role of hydrogen peroxide in the redox signaling in human body? And if you look at the hydrogen peroxide reactivity, I have shown a picture here. If you open this hydrogen peroxide bottle by bare hands without gloves, you see something like this. It actually oxidizes the skin proteins. So the skin proteins gets oxidized and it becomes white like this. But if that is the case, how does the level of hydrogen peroxide, how is that is maintained in human body? And if you search for the role of hydrogen peroxide in biological systems, you find about 125,000 papers dealing with the reactions involving hydrogen peroxide, the biological functions involving hydrogen peroxides, with millions of citations. And hydrogen peroxide production, protection, and signaling action, very important to understand these aspects to develop any synthetic enzymes that can control hydrogen peroxide in the human body. And hydrogen peroxide oxidizes cysteines on proteins to initiate the redox biology. If you look at this slide, when we have about 4 to 5 grams of iron in the human body, When we know when iron is kept outside, iron rod is put outside, moisture, water, oxygen, we see a rusting that takes place. But human body has about 4 to 5 gram of iron. Only 1 milligram is lost per day, but we don't see any such rusting in our human body. If rusting takes place, it leads to diseases like neurodegenerative diseases, Alzheimer's, Parkinson, and so on. So the iron in biology is tightly regulated. And why? Because the hydrogen peroxide can react with iron to form hydroxyl radical that can damage DNA and then can induce lipid peroxidation, and this we call it as oxidative stress. The positive side of reactive oxygen species, hydrogen peroxide, which is produced from superoxide by superoxide dismutase, can oxidize protein thiols, and the protein thiols or thiolates is oxidized to protein sulfenic acid, and that initiates redox signaling. And there are many enzymes like peroxide redoxins, glutathione peroxidase, catalases. These are involved in controlling the level of hydrogen peroxide in human body, and this is called redox biology. When we look at the reactive oxygen species function in redox signaling and oxidative stress, if you look at this slide, the self-renewal of healthy stem cells, differentiation of cells happens by reactive oxygen species, and when the level of reactive oxygen species goes down in the cells will not be able to self-renew, and excess amount of reactive oxygen species lead to stem cell exhaustions. So the lifespan depends on the level of reactive oxygen species, and if you look at the basal level of reactive oxygen species, which is kept slightly lower than the highest optimum level. So if you increase the reactive oxygen species level in the human body, actually the lifespan increases, but nature chose to set that lower than the optimum level of reactive, the highest level of reactive oxygen species. And then if you increase the reactive oxygen species level further, the lifespan decreases. You can see the right side of the curve here, the lifespan decreases. And that's the reason why the basal level is lower than the maximum level, because in the biological systems, increase in the reactive oxygen species can be taken care by the antioxidant enzyme system in the human body. And we are looking at the two sides of the same species. In most of the biological processes are like that. And hormones, for example, reactive oxygen species, the positive side is that stem cell renewal, the left side of the one I have shown here, proliferation, differentiation, healthy immune responses, and longevity. If you look at the negative side of the reactive oxygen species, stem cell exhaustion, tumorigenesis, autoimmunity, and senescence. So the redox biology encompasses both the physiological and pathological role of ROS, determining whether to use pro-oxidant therapy to promote ROS or antioxidant therapy to prevent ROS, that remains central questions in redox biology. So what we are trying to do is that, can we control the reactive oxygen species, oxidative stress related diseases? I have listed few of them: kidney diseases, brain, heart, lung, joint, multi-organ, vessels, and eyes diseases. All of them are associated with oxidative or nitrosative stress. So uncontrolled production of reactive oxygen species and the system's inability to take care of the reactive oxygen species lead to an imbalance, and which is called oxidative stress. So we are working on a particular enzyme called glutathione peroxidase, and this is a very interesting enzyme. It uses selenocysteine, which is known as the 21st amino acid, in the active site to convert hydrogen peroxide to water without producing any reactive oxygen species further or molecular oxygen. And the GPX deficiency directly induces an increase in the oxidative stress. If you look at, for example, the selenocysteine, the reactive moiety, which has a pK of 5.2, which is much lower than the pK of cysteine or the serine, which is 13. And this is the reason why nature selected selenocysteine, which is unusual amino acid, as part of these enzymes. So We have been working on small molecules that functionally mimic these enzymes, not only in the test tube, but also in the living cells. This is a compound known as Ebselen. You might have heard about it. This was reported to have GPX mimetic activity in 1984 and undergone several clinical trials. But if you look at this structure, which is a cyclic 5-membered seleninamide structure, this has several biological activities: antioxidant, anti-inflammatory, antimicrobial, free radical scavengers. But if a compound shows multiple activity in human cells, there is a problem with it. And this belongs to a compound called isothiazolones, the second structure shown here. So isothiazolones are one of the paints, which is pan-assay interference compounds, and hundreds of chemical compounds fall into this category because they can covalently modify proteins. So they react with the chemically reactive proteins and non-specifically and non-drug-like ways. And Ebselen is quite similar to this. The difference is that sulfur is replaced by selenium. So one would wonder how Ebselen has undergone multiple clinical trials for various diseases. These are biological effects of Ebselens, and this is Ebselen as a drug. You see title like this, the weirdness of Ebselen. And looking at the structure, people say that it's not a drug because it can bind non-covalent, it binds covalently to proteins. But you see also reports like this. Human trials suggest rescued drug could be safer treatment for bipolar disorder. A drug destined for scrap heap has been rescued by Oxford scientists who may have found it a new role in treating bipolar disorders. Ebselen is part of the NIH clinical collection, a chemical library of bioavailable drugs considered clinically safe but without proven use. You might have come across this paper recently for COVID-19. Published in Nature, accelerated article, this compound inhibits a protease from a COVID-19 virus, and the screening of 10,000 compounds identified Ebselen as one of the 6 compounds potential COVID-19 therapeutics with the IC50 value of 0.67 micromolar. And when we started looking at structures of the GPX mimetics, Ebselen is, of course, the well-known compound has undergone several clinical trials. We made a series of compounds shown on this slide, diselenides with the tertiary amide, secondary amide, secondary amine, tertiary amine with an additional methoxy group here, and we performed a structure activity correlations, and we select compounds which are more potent and then try to do studies whether these can functionally mimic enzyme or substitute these enzymes in mammalian cells when these enzymes are inactivated. So we started studying the reactivity of these compounds, understanding the mechanism of these compounds, and we found that Ebselen, the major problem with Ebselen is the reactivity, which is known as the thiol exchange reactions. If you look at the selenosulfide bond here, the selenium is more electrophilic than sulfur. So therefore, the electrophilic glutathione should attack at the selenium center, not at the sulfur center. Therefore, you often see a thiol exchange reaction happening at the selenium center rather than generation of a selenol, which is happening in the protein. And this was the major problem that we identified with Ebselen. But there is also a positive effect, positive side of Ebselen. Ebselen is extremely low toxic. And although people think that selenium compounds may be toxic to human cells, Ebselen is not toxic. The reason is that Ebselen undergo a redox cycling. You can see the selenic acid here, EOH, which eliminate a water molecule by reacting with an amide backbone here, forming a cyclized compound. So if you put in a cells, the Ebselen selenium nitrogen bond is cleaved by thiols, But after some time, what happens? It undergoes reversible cyclization, which protected the selenium moiety from irreversible oxidation. And irreversible oxidation, you can see here in the case of PTP1B, there were two papers in Nature in 2003 about the reversible cyclization of a sulfenic acid in the protein. And we have shown the seleninic acid in this Ebselen also behaves similarly, and it protected the active site. And what we found was, I'm skipping all our studies and 10-15 years of studies, which led to believe that the carbonyl group on Ebselen is creating a problem. So we wanted to remove the carbonyl group, but we want to have the cyclized compounds. So you cannot cyclize this compound if you do not have the carbonyl group there. It just forms an open chain diselenides like this. So what we did, we introduced a methoxy group. You can introduce any groups there, forcing the selenium to cyclize. And that is a crucial step in this process. You introduce a steric hindrance here, which forces the seleninic acid, which is produced from these compounds, cycle process. So we have developed this 4A compound, which also prevent DNA damage, both single strand and double strand damage. You can see the hydrogen peroxide mediated DNA damage, and with the compound, it brought back to the normal level. And what we found, the key difference between Ebselen, which people spend millions of dollars carrying out the clinical trials, still it is safer drugs in mammalian, safer compound to test in mammalian cells, and someone should identify a disease to treat, and COVID-19 is one of them, recently identified as a target for Ebselen, but there may be other possibilities for using Ebselen as a drug. But what we found is that irreversible inhibition of mammalian enzymes which have cysteine residues in the active site. One of the enzymes that we studied was human glutathione reductase, which is an NADPH dependent enzyme which converts glutathione disulfate to glutathione. So therefore, this is a key enzyme that maintains redox balance in the cells. And what happens, Ebselen goes and binds to the cysteine active site and blocking the activity irreversibly. What does it mean? It increases the glutathione disulfide level in the cells, deplete the glutathione. So all the redox processes, including the redox biology, the reversible redox signaling I talked about, is affected by this. And the compound that we developed, removing the carbonyl groups, what we find is that it doesn't inhibit glutathione reductase. And that is safe. Actually, redox state of the cell is maintained by these compounds. So therefore, a structure activity correlation studies led to the development of a cyclic selenozoles that can actually take care of the oxidation state, oxidative stress in the cells, but without affecting the redox processes. And when we do these studies in vitro in the test tubes in the lab, showing that these compounds functionally mimic the glutathione peroxidase, it is not sufficient. We have to also show that they do not have any negative effect affecting the redox cellular processing process. So next few minutes, I will quickly go through the other type of compounds that we are interested in. So we are interested in set of compounds called nanozymes. So nanozymes are nanomaterials that functionally mimic natural enzymes. And nanozymes as artificial enzymes are attaining a greater interest recently. So our early studies focused on vanadium pentoxide nanowires as artificial enzymes. As you can see here, the vanadium pentoxide nanowires convert hydrogen peroxide to water without generating any other reactive oxygen species or molecular oxygen using glutathione as cofactor. So this functionally mimic glutathione peroxidase, the first non-selenium based compound or the nanomaterial to show glutathione peroxidase activity. So we have studied the activity in the cells, and you can see here the untreated cells, the cells treated with vanadium nanowires, no toxicity, and the cells treated with hydrogen peroxide, it enhances the reactive oxygen species level, and then vanadium nanowire controls the reactive oxygen species, brings back to the redox state to the normal level. In contrast, the copper sulfate, which is known to generate reactive oxygen species, are not able to rescue the cells. And we can also see here that vanadium complexes like this one, which has a peroxo-vanadium, oxo-vanadium complexes, and they are highly toxic. Although both of them have vanadium in +5 oxidation state, just like our vanadium in the nanowires, and also bulk V2O5 is toxic to the cells. You can see the bulk V2O5 reduces the cell viability. Vanadium nanowire, there is no change in the cell viability. But when you use the vanadium complexes, which also have vanadium in the similar oxidation state, they are toxic. And what we found is that when the vanadium +5 is present as part of the nano surface, we find that the oxidation state is quite stable. But by treating that with glutathione, we are unable to reduce the vanadium 5 in the case of vanadium nanowires, but vanadium complex is readily reduced by glutathione to produce vanadium in 4 oxidation state, which can be easily monitored by EPR. So what we found is that nanostructures act significantly different from small molecules and bulk materials. And by exhibiting glutathione peroxidase type reaction on the surface without permanently changing the oxidation state of the metal ions. And this concept led to the development of further nanomaterials. We have shown that the activity is size and morphology dependent, and V2O5 having different morphologies show different activity. So you can see here the different morphology showing different activities. We do not see any correlation with the surface area, but we see a correlation with the type of planes or the facets which are available on the surface for the catalytic reactions. And we found that the formation of a vanadium peroxo bond from vanadium oxo by hydrogen peroxide is the rate limiting step. Once the peroxido is formed, then it's cleaved by glutathione by attacking at the oxygen center here, and then the reaction continues. And we have done time-dependent in situ FT Raman spectroscopy to find out that this is the rate limiting step of these reactions. And we have shown that different type of planes, 001, 1010, 100 planes, and they have shown different activity on the surface, and we find that it is morphology dependent, facet dependent activity of these nanozymes. And we also developed an Mn3O4 nanozymes with multi-enzyme activities. This nanomaterial mimic the function of 3 enzymes: superoxide dismutase, catalase, and glutathione peroxidase. The 3 enzymes I talked about in the beginning, and they functionally mimic those enzymes to control the total level of reactive oxygen species. And we have shown that the reactive oxygen species scavenging ability actually prevent the morphological changes that takes place in SHSY5 cell lines. And this one particularly mimicking the triple enzyme activity, 3 enzymes, superoxide dismutase. So this material converts superoxide to hydrogen peroxide and hydrogen peroxide to oxygen water by mimicking catalase, and by mimicking glutathione peroxidase, it converts cleanly the hydrogen peroxide to water by using glutathione, and this works very well in the cell. So that means they are taken up by the cells and function as an enzyme. And next 2 to 3 minutes, I talk about superoxide and endothelial dysfunction. So where are we heading to? Can we use this material to regulate the redox signaling in the cells and control the reactive oxygen species and control diseases? When we talk about the superoxide generations, in the resting state, NADPH oxidase is like this. But when it goes to active state, NADPH is oxidized to NAD+, which releases 1 electron. And that 1 electron is pumped across the membrane, and then superoxide is reduced by 1 electron. Molecular oxygen is reduced by 1 electron to generate superoxide. And just this 1 electron reduction, you just look at the arrangements, several phosphorylation reactions, and huge transmembrane protein, which controls how efficiently or how controllably this electron is transferred, and then 1 electron reduction is performed. So therefore, the 1 electron reduction is done by very controlled way in mammalian systems. But what happens if enzymes can generate superoxide on its own without involving any such very phosphorylation, dephosphorylation reactions arrangements and the regulated reduction of molecular oxygen? One such enzyme is nitric oxide synthase. We all know about it. It's a metalloprotein which generate nitric oxide from L-arginine by two-step reaction. So in the first step, it converts arginine to N-hydroxyl L-arginine, and the second step, it converts to L-citrulline, and in this process, nitric oxide is released. And this is a huge enzyme which has multiple domains. The reactive domains is a heme where the reaction takes place, and it takes electron from a tetrahydrobiopterin and the reductase domain, which is actually supported by another protein called calmodulin, which is a calcium dependent enzyme. And this is the endothelial nitric oxide synthase. So the electron goes from NADPH, FAD, FMN, which is called the reductase domain, to the oxygenase domain, which is a heme protein. And then the reaction is performed to release NO. And a very, very interesting enzyme. And it has two steps. In the first step is called arginine conversion to N-hydroxyl arginine. And many of bioinorganic chemists are very familiar with these steps, where Fe3 plus is the native form of an enzyme, takes up one electron. In this case, it is from NADPH, from the reductase domain. It's reduced to Fe2 plus, and we know that low spin to high spin then reduction takes place. Then what happens? The Fe2 takes up oxygen, which is similar to hemoglobin oxygen binding. It binds and forms a superoxide iron 3 plus complex. In the case of oxygen binding, it is reversible, where oxygen is released after superoxide formation. That means one electron from Fe2 plus migrates to oxygen, forms a superoxide, but superoxide is still bound to the metal center. But what happens if the superoxide gets released from the metal center? Let us assume that hemoglobin is dysfunctional or myoglobin is dysfunctional. The superoxide gets released from that. It leads to diseases. It's a very, very dangerous process. And in the case of endothelial nitric oxide synthase, what happens is that the superoxide gets released from this if there is no electron donor. In this case, it is tetrahydrobiopterin, which is electron donor, and that donates one electron to convert the superoxide to peroxide on ion, and that conversion does not take place because there is no electron donor, and this leads to superoxide release from nitric oxide synthase. So the second step to produce nitric oxide doesn't happen, and this generates superoxide instead of nitric oxide. And this is, we study as a shunt pathway, but shunt pathway leading to diseases. And endothelial nitric oxide synthase, nitric oxide is important to maintain the endothelium. Endothelium is the inner lining of the blood vessels. So the normal arterial wall has the endothelial cells in a properly functional state inside the endothelium. But when onset of endothelial dysfunction happens, you can see the atherosclerosis and then myocardial infections and lead to heart attack or stroke, that is complicated lesions and fat accumulation and damage to endothelium takes place and blood flow is completely blocked. And nitric oxide plays very, very important role in maintaining the health of the cardiovascular systems and the blood vessels, which actually transport, the blood. And we looked at the nanozymes and small molecules, whether they can regulate the nitric oxide level in endothelial cells. And doing experiments in primary cells is much more difficult than using cell lines. Cell lines are commercially available, you can buy and use it. But to study the nitric oxide, we need to isolate the endothelial cells, study in the primary cells as soon as they are isolated. And it's quite difficult to do. But what we did, we used Mn3O4 to regulate the formation of nitric oxide, and by controlling the superoxide level. We know Mn3O4 functionally mimic the superoxide dismutase. Therefore, one can control the level of superoxide. Therefore, the reaction between the superoxide and the nitric oxide, which is generated by endothelial nitric oxide synthase, can be regulated by this nanomaterial. So this is one slide I just want to show that we are able to regulate the nitric oxide levels. The first slide is the superoxide. First plot is the superoxide ones, you can see using nanoflowers, manganese nanoflowers, Mn3O4, we can bring back the superoxide level to the controlled, untreated cells. And we can also bring back the nitric oxide level. We use the DAFF, a specific dye for nitric oxide in living cells. And we have brought back the nitric oxide level to the control level, which is induced by hydrogen peroxide. We could see that similar level in the presence of Mn3O4. And this is very interesting. Using an enzyme mimetic or artificial enzymes, we can control the NO production in these cells. And we also, what we did, we inhibited the enzyme with dithiocarbamate, which is an inhibitor of SOD1, which completely inhibit the superoxide dismutase. After inhibiting the superoxide dismutase, if we supplement the cells with Mn3O4, we are able to substitute the enzyme activity with this nanomaterial. And you can see the morphological changes that takes place when you inhibit superoxide dismutase with the DTC. The cells are dying and the morphology has completely changed. These are endothelial cells, the primary endothelial cells. And you can see that it's restored back with treatment with Mn3O4. And what we are trying to see is that we are trying to develop a general strategy. And this is my last slide, which talked about oxidative eustress and oxidative distress, because some stress is good. And oxidative stress is good, which is important. As I have shown in the introduction, actually it increases lifespan, increasing the reactive oxygen species a little bit. This is the slide I have taken from a paper by Professor Helmut Sees, who is considered as a redox pioneer, who talks about the hydrogen peroxide level outside the cells, extracellular and the intracellular. The cellular responses, if they are monitored, you can see the intracellular going from 0.01 micromolar to 10 micromolar. You can see how the cellular function changes. At lower concentration of the reactive oxygen species, which I talked about the redox biology, proliferation, migration, angiogenesis, all this take place. When you increase the concentration of the reactive oxygen species, the stress responses, adaptation like NRF to gene expressions and associated gene expression takes place. This is the stage where we call it oxidative eustress. But when the oxidative stress goes further, reactive oxygen species level goes further, which is known as oxidative distress, inflammations, fibrogenesis, tumor growth, metastasis, growth arrest, cell death take place. And a distinction of oxidative eustress and oxidative distress may occur at a fine borderline. And using artificial enzymes, finding this fine borderline, can we control the redox signaling in the cells by using artificial enzymes when the natural enzymes are inactivated by oxidative stress? So that is the entire focus of my group, and talking about controlling the redox signaling by using artificial enzymes. So this is one of the area that we work on. We also work on other areas like expanding genetic code with unnatural amino acids. We work on thyroid hormone metabolisms and improvement of cellular uptake by different strategies. Recently, we have shown that a halogen bonding can be used as a strategy to increase the membrane transport of small molecules and also proteins. So I don't have time to talk about all those. With this, I would like to thank my former students who contributed a lot to this work, and current PhD students working in different areas, and the RES postdocs. Two of them I highlighted here worked extensively on controlling the reactive oxygen species in mammalian cells and controlling the nitric oxide level in endothelial cells. And I thank my collaborators, and this is the website. If any of you would like to know more about our activities, there is an publications on our work. Please look at our website.